Okay, so we'll start a discussion of the special senses. That'll be taste, smell, hearing, balance, vision, and we'll really just touch briefly on olfaction and gustation, and then we'll go on to vision and hearing and balance. So let's start quickly with olfaction, which is smell. We talked about this a little bit in class the other day, but olfaction occurs when a molecule, an odor molecule from the air, comes in through your nose and gets dissolved in the mucus that's produced at the roof of your nose. If you can kind of look at this picture here. So you can see the odor molecules here are going to come, and there's this kind of yellow layer here of it's like a mucus layer. And the particles are going to kind of dissolve in that, and once they dissolve, they can then land on the receptors. So they'll land, for example, on a receptor here. And that's going to trigger a response in this cell that's going to eventually create an action potential that travels through the cribriform plate, through the holes in the cribriform plate, you see there, and up to the olfactory bulb. So the axons leave through the olfactory nerve, and then they become the olfactory bulb, which we could actually see grossly on the sheep's brain that we looked at last semester. And then those axons are going to actually synapse directly with the cerebrum at the olfactory cortex, like we talked about in class earlier today. And so it has a direct connection. It doesn't stop in the thalamus or the midbrain or somewhere else like pretty much all of the rest of our senses do. And that kind of gives a quicker, a more powerful connection between smell and the rest of our brain because it directs connectly. It also sends some information to the limbic system as well, and that's why, as I was mentioning in class, a smell can unlock a memory that you've had from before, or unlock an emotion that can even surprise you. I gave this example to the class last year, but when we were away at Africa for, it was about 2.5 years we had been gone, had not been back to America in that time, and we came back and it was around this time of year. And you know how you walk into somewhere like Target or wherever, and there's just pumpkin spice everything. There's pumpkin spice latte at Starbucks, and there's pumpkin spice candles and cookies and everything. And so the smell of pumpkin spice is kind of everywhere. And I shocked myself when I walked into Target one day, and I got hit by that pumpkin spice latte smell coming from the Starbucks, and tears came to my eyes, and I was surprised that I was getting so emotional from the smell of pumpkin spice latte, but it just kind of triggered in my memory all of the things that I loved about this time of year, the cool weather and the leaves changing and the crunch of leaves under your feet and the different smells and flavors, and we had missed that for a couple of years, and so when I first encountered that again, it really kind of awakened all of these emotions. And you'll see that smell can do that. Smell is a very powerful sense in that regard. Taste is, of course, closely related to smell. So if you don't smell well, you don't taste well. And you've probably experienced that when you have a cold. So the two of them are together. You need to be able to smell well in order to taste well. But if we look separately at taste, we call that sense gustation. And it's the same idea where taste molecules first have to dissolve. That's one of the reasons we have saliva. It actually helps us taste things better. There's lots of other benefits of saliva, but that's one of them. And then if we look kind of in on this, if we zoom in on this picture here, we'll see that those dissolved particles, so let's say you ate something and it's dissolved in the mucus, some of them are going to actually go down into what we call a taste pore. And that taste pore, so on this picture, we would kind of have that taste pore would be here. And it's going to land on, whichever of these receptors it fits. And so, for example, if it's sour, if it's has maybe protons in it. That's what makes things taste sour, acidic. Then it would land on this sour receptor, and it would stimulate that, release neurotransmitter, and these, which would cause an action potential here on these sensory neurons, and then they would go to the gustatory cortex in the brain, and then you would know that you're tasting something sour. Same thing would happen with bitter, with sweet, and I'm not sure if we talked about umami in class in anatomy, but umami is kind of a newer flavor that's been discovered maybe in the last 10 or 15 years. And I remember hearing about it, watching all these food shows, 10 years ago, and thinking that maybe they were trying too hard. These food people were kind of making up a new flavor, and they were really just trying too hard to come up with something new. But it's actually been studied physiologically, and they have isolated the taste receptor for umami, and they found that it's stimulated by glutamate. So foods that have glutamate in them will land on this umami receptor, and then will stimulate that receptor to release neurotransmitters onto the sensory neurons, and then they will carry that signal to the brain, and the brain will say, that came from an umami receptor, that must be fish, or that must be bacon, or that must be ketchup, along with the other flavors that it's combined with. So that's kind of how taste occurs. And the gustatory cortex is in the insula, which is kind of just deep to the temporal lobe, and that's where all of those receptors land. Okay, let's start our discussion of vision with a case. We'll call this the case of Fatima. So Fatima is a 45-year-old female, and she came to the hospital complaining of a headache, fatigue, and that she's been losing her peripheral vision for about the last 3 months. So at first it was barely noticeable, but now she can clearly tell that she cannot see in her periphery. She feels like she has tunnel vision. And here's an MRI of her brain to kind of show you where the problem is. Now, I'm not going to tell you where the problem is yet. We're going to go through learning how vision works, and then we'll come back to this case at the end and see if we can figure out in this picture of the MRI where the problem is, what's going on with Fatima's vision. Okay, so vision is basically just the interpretation of photons. So photons, or rays of light, are bouncing off of objects all around us, and those photons eventually come through the eyeball and they land on the retina. And then the retina produces action potentials that go through the optic nerve. And then from the optic nerve, we see that they cross. So if we see the retinas in here and in here, and light is bouncing off of objects, the action potentials are then going down the optic nerves, you can see. Notice that they cross at the optic chiasm, and we talked about that in anatomy. That optic chiasm that sits kind of right in front of the sella turcica where the pituitary gland is, if you remember that area. And that crossing is important for binocular vision, and we'll talk about that towards the end of the lecture. We can see that some of the information goes, as we mentioned before, to the midbrain, and that's going to help coordinate eye movement so that your eyes move at the same time, so that you can track an object at the same time and not have double vision. And then the rest of those fibers go back to the occipital lobe where they can be interpreted. So let's talk about how light passes through the eyeball, because before light can hit the retina, which is actually the sensory organ of the eye, the retina is really the sensory organ. It's what's receiving the photon of light and leading to an action potential. That happens at the retina. But you have a whole lot of eyeball before you get to the retina, and those photons of light have to pass through several regions of the eye before they can hit the back of the retina and actually produce an action potential that can be perceived as vision. So the first place that light, so if we have light coming in, the first place it has to pass through is the cornea. And that cornea will actually bend the light. So light that comes at one angle will be bent when it hits the cornea. And about 2/3 of the bending of light will occur at the cornea. And we call the bending of light, we call that refraction. So 2/3 of refraction occurs at the cornea. Okay, then between the cornea and the lens, remember, is the anterior cavity, which is composed of the anterior and posterior chamber. So that's kind of this area in here. So the light then has to pass through that chamber. And the next place it's going to pass through is the lens. And the lens does the final third of refraction, or the final third of the bending of light. So light that passes through the lens will be bent again. And then that light goes through the posterior chamber and pick up that light ray here from another angle. So remember, the light's going to come through here, go through the cornea, anterior cavity, lens, posterior cavity, until it eventually lands back on the retina. And when it hits the retina, that's what actually triggers the action potential that stimulates the optic nerve that travels to the brain, and you actually can perceive the light. Okay, now this idea of refraction is really quite important. So if we kind of look at an eyeball like this, and we'll say that this here is the cornea, and then let's put a lens in here, and we'll have the retinas kind of back in there. So as light comes off of an object, so let's say we have an object here, and we want to look at the tip of a pin. So light is going to come off of that tip, and it's going to hit first the cornea, right? And when it goes through the cornea, remember, it's going to be bent. We call that refraction, that bending of the light. Then it's going to enter the anterior cavity, and it's going to hit eventually the lens. So we'll have that be the lens. And as it goes through the lens, it gets bent just a little bit more, until eventually the goal is for all of those rays of light to converge together at one spot on the retina that corresponds with the spot that you saw in space. So you've got two sites where that light gets bent, and that bending of light is important so that the rays of light that are bouncing off of that object can come back together and hit the same spot on the back of the retina. And we'll see why this is important here in a minute. But remember, the cornea is kind of, it's fixed. It's not something that your body can just change the shape of when it needs to. There's no way to change the shape of your eyeball with muscles or anything like that. Now, sometimes squinting helps change the shape of the eyeball a little bit, and this is why we squint, in order to see things a little bit more clearly. But there's no really mechanism in place to do that naturally. So if your eye is trying to focus on something far away or on something near, it has to bend the light to different degrees. So things that are coming at a distance, light rays that are traveling from a distance, they require less bending, and objects that we're looking up at close range, they require more bending of those light rays. And so we have to have a mechanism for bending that light. And even though 2/3 of the light is bent at the cornea, there's no way to change that bending. So the only place that we can really affect the refraction of light is at the lens. So the lens, if you remember, has that ciliary muscle around it, and it can constrict and relax and can make that lens either flatter or more concave, and that's going to help bend the light more or less to help us focus on things that are near and focus on things that are far. So when things are near, we want a more rounded lens, a more concave lens, and that will bend the light more. When we want less bending, so when we're looking at something far away, we want the light to be bent less, and in that case we want to kind of flatten out the lens, which will be less bending of the light. And we're going to see some examples here in a minute. So let's go through this. So let's say we're looking at something nearby. This kind of looks like the picture that I drew. You've got these these rays that are coming out at quite an angle. And so if you didn't bend that, if you didn't bend those rays, right, they would not land together, they would land separately, and you would have, you know, several areas that were being stimulated at the same time, and your brain would interpret that as seeing multiple versions of that object all close together. Your eye might interpret that as blurry vision. And so it's important to bend those rays so that eventually, as you see here in the picture, they come through the cornea and they bend. Now they're a little more parallel. And when they get through the lens, they have to bend again so that eventually they all fall in one point on the back of the retina. We call that the focal point. And ideally, we would like to have the focal point be on the fovea. Remember, the fovea is the part of the retina that's in the center of the macula, and the macula is the area where you get the sharpest vision. So the center of that macula is the fovea, and that's where the absolute best vision occurs. So if we really want to see something clearly, we want to focus those light rays onto the fovea of the retina. And we do that by either making our lens flatter or making our lens more rounded, and that can bend the light more or less. So for near viewing, we want a lens that's a bit more rounded because we need the light to bend more because the rays that are coming off are more divergent. On a distance viewing, you can see that by the time the rays actually reach our eye, they're already pretty much parallel. And so we don't need to do quite as much bending. And remember, we're already going to get 2/3 of our bending at the cornea, and there's really nothing to be done about that. So when we get to the lens, we don't want to bend it too much. If we bent it too much, we would then get these rays once again kind of diverging and giving us multiple spots on the retina, which would lead to blurry vision. So we don't want to overbend the light. We want to bend it just enough so that in the end, the rays bend and they converge together at the fovea. And this is having the ciliary muscles relaxed, allows it to flatten out. So near vision, you're going to have the ciliary muscles contracted. You're going to have a more rounded lens. You're going to have more bending of the light. Distance viewing, you're going to have the ciliary muscles relaxed, which is going to cause the lens to flatten out, which is going to lead to less bending of the light. Remember, that bending of the light is called refraction. Okay. Just of interest, really not a big deal, but just so you're aware, because of the way light bounces off of objects and moves through the eye, the way that the objects land on the back of the retina is actually upside down. And so when the brain actually receives the action potentials, it receives the image upside down, and then our brain is able to flip that right side up so that we can see things as they really are. I want to talk about a few of the problems that we have with vision. The first is called myopia, which is nearsightedness. And we use this term myopia or myopic, even talking about kind of philosophically. We might say, oh, don't be so myopic. That means don't be so concerned with what's right in front of your face that you forget to see, the future. You forget to see what's beyond your circumstances. So when we're myopic, it means we're short-sighted. It means we're not really paying attention to what's going on in the distance. In a physiological sense, what it means is we can see objects up close fairly well, but when we try to see things in the distance, they become blurry. And that's most people who wear glasses, especially if any of you wear glasses. Most of you are nearsighted. Personally, I'm also nearsighted. That's why I wear glasses. Some of you might wear contacts. Same idea. It's because your distance vision is blurry. So we say you're nearsighted because you can see things near, but you don't see well far away. So we call that myopia. And what happens here is the shape of your cornea is incorrect. And because it's bending the light too much, so there's too much of a curve here in the cornea, that's causing the light rays to bend too much. And what happens is those um, light rays, they end up converging at a point that's in front of the retina. So when they actually hit the retina, you can see that they're kind of scattered. So you don't get one clear point where all the light is landing. You get that light kind of scattered along an area within the retina, and the retina sees that as multiple points of the same object. So it looks to the retina like you're seeing, 10 of the same object. But those 10 objects are so close to each other that your brain doesn't distinguish each individual object. Your brain just perceives that as blurry vision. So what we need to do to correct that is we need to find a way to make the light bend less, so that when it finally converges, it will converge back here on the retina. So let's look at how glasses do that. So glasses, most of us are wearing diverging lenses or concave lenses. So that means, let's say we're looking at something that's far away, right? So if we look at this picture here, remember that distance viewing, by the time the rays land on your eye, they're pretty much parallel. And remember, they were converging too soon, those light rays, and that's why we were getting our blurry picture. So the way that diverging lenses help is they actually cause the light rays to diverge slightly. Instead of being parallel, they cause them to diverge before they hit the cornea. So then when they pass through the cornea and they're refracted, they become parallel again. And then when they go through the lens is able to bend enough to focus those rays back so that the focal point lands on the retina. And then you see one clear image. So it's all about getting the light rays to diverge before they hit the cornea, so that the overbending of the cornea really becomes a non-issue because you have caused those light rays to diverge before they hit the cornea. Another way that we fix this, so you can do it with glasses or contacts, and that's what a lot of us do. But there's also something called Lasik surgery, and I'm sure you've all heard of it, and some of you have probably even had it. And this tries to attack the problem at the eye itself. So the problem for most of us who are nearsighted is that our corneas are misshapen. There's too much concavity, too much of a curve to our corneas. So if we could find a way to flatten out that cornea, then that would cause the light to be bent less, and it would not converge too soon. It would converge where it's supposed to on the back of the retina. So the way LASIK surgery does that is they actually cut a flap, you can see here in the cornea. Then with the laser, they actually flatten out that cornea. Then they put the flap back over, and you can see that you went from kind of a curved cornea here to a slightly more flat cornea. So that way when the light comes in, it gets bent less than it did before. And it's fairly successful. Your book talks about a 70% success rate. I think it probably depends on who's doing the surgery and what technique they're using. But I've seen and heard of higher success rates even into the 80s. Some people do still have to wear glasses for close-up reading because this doesn't fix that problem, but it does help a lot of people who have problems with distant viewing. Okay, so the opposite of myopia would be hyperopia, or in kind of plain language, we would call that farsightedness. That means we can see fine when we look out at a distance, but we have trouble reading things that are close up to us. And so if we look at the picture here, if we're trying to look at things that are far away, remember that's going to be fairly parallel light rays coming in. And again, because of the shape of the cornea, maybe in this case it's a little bit too flat, so we don't get enough bending of the light. So by the time it hits the lens, even though the lens may try to do maximum bending, maximum curvature, it's still not able to make those rays land together on the retina. You see when they land on the retina, they're actually separated. So again, you would get a blurry vision because you're going to get multiple spots activated along the retina instead of one spot altogether. So this is kind of the opposite problem that we had in myopia. This happens a lot as we get older, and it's not really so much a problem with the cornea as it becomes a problem with the lens at that point. So people who get farsighted later in life, we call that presbyopia. Presby is kind of a word for old or elder. Again, comes from a Greek word. And so it happens to most of us as we get older. Like 50% of people after age 40 get it, and the number just goes up every decade after that. So you can pretty much plan on having some farsightedness when you get older. And again, the problem there isn't so much with the cornea as it is with the lens getting stiff and not able to contract down enough to bend the light quickly enough so that it lands on the back of the retina. So you can imagine we're going to want kind of the opposite fix for that. So we're going to, instead of using a diverging lens, we're going to use a converging lens. So that's going to cause the rays of light to actually, instead of being parallel, to be slightly converging already by the time they hit the cornea. And that way the lens is able to bend it the rest of the way so that it lands together in one point on the back of the retina. So converging lenses or a convex lens will help hyperopia. Okay, so once the light finally hits the retina, now we're really getting to the physiology. So this is where we're actually going to create action potentials through the release of neurotransmitters, and that's going to send the electrical signal to the brain that can then be interpreted as something that we see. So the photoreceptors are we've been talking about different kinds of receptors on cells. So a photoreceptor is a receptor that responds to a signal sent by light. And so these are actual photons of light that are hitting these receptors. And in common language, we call these receptors rods and cones. So they have to receive the light. So the light has to literally land on these receptors, these photoreceptors. And then they will cause a change inside that cell that leads to a change in neurotransmitter release that leads to a change in action potentials. So the first step is called photoreception. That's the light landing on the rod or cone. And then the change from photon to electricity, we call that transduction. So taking that photon of light and turning it into an electrical signal that can then travel to the brain, we would call that transduction. So there's a molecule inside the rods and cones, and this molecule is kind of the key to receiving that photon of light and causing that change in neurotransmitter release. And we call that molecule rhodopsin. And rhodopsin is partly made from vitamin A, which is why there is some truth in talking about carrots and things like that are good for your vision. For most of us, we get enough vitamin A. We probably don't need extra carrots, but there is vitamin A in carrots, and vitamin A is used to make rhodopsin, and rhodopsin is used to create vision. So rhodopsin will receive the light, and it actually changes shape, and that change in shape of the rhodopsin actually leads to a change in neurotransmitter release, and the change in neurotransmitter release will cause a change in action potential, and that's what the brain is actually sensing. So let's kind of go through the steps and see how that works. Now, there are different kinds of photoreceptors, cones and rods, of course, but some of them respond more to dark and some more to light. But kind of the basic photoreceptor that we'll talk about is this resting dark. We might call them darkness receptors. And so what that means is when there is no light, those photoreceptors are releasing more neurotransmitters. So they're actually more active in the dark. And that's because there's an ion gate that's open. These sodium channels are open when the rhodopsin is not affected by light. So the shape of the rhodopsin in the dark actually causes the sodium channels to be open. Sodium is coming into the cell and producing action potentials and those action potentials are releasing neurotransmitter. So in the dark, you actually have more neurotransmitter release and more action potential. As light hits the rhodopsin, however, you can see that's the part of the molecule they want to show you changing. So in the picture, it goes from bent to straight. And that change in shape actually closes the sodium gate. So sodium is no longer able to enter the gate. That reduction of sodium entering the cell results in a reduced frequency of neurotransmitter release and a reduced frequency of action potentials. So there are cells that are synapsing with the rods and cones, and we call them bipolar cells, and they're the ones that are actually sensing the neurotransmitter that's being released from the rods and cones. And so those neurotransmitters are causing action potentials in the bipolar cells, and those action potentials are then going on to other cells called ganglion cells, and from there they all bundle together into the optic nerve. So if we kind of review what we just went over, when it's dark, the sodium channels are open. You're getting more sodium entering the cell, so you're getting release of more neurotransmitters from the rods and cones onto the bipolar cells, and you're getting an increased frequency of action potentials sent down the optic nerve. When photons of light hit the rhodopsin, change the shape, sodium channels close, less neurotransmitter is released to the bipolar cells, so there's a reduced frequency of action potential that travels down the optic nerve. And so it's the change in action potential that the brain is actually receiving from the optic nerve. Okay, just a few quick words on rods and cones. Rods are by far much more numerous in the retina. They're kind of the workhorse of vision. They help us to see shades of gray. Shades of gray. They help us to kind of see a contrast between different objects that we're looking at, and it's what we use in nighttime vision when light is low. Cones, on the other hand, are better for color vision. So cones are much fewer, but they're better, meaning they give us a clearer vision. They can see colors, for example, and help us really know what we're looking at. The fovea, which we mentioned earlier, that's the central part of the macula, the place where the best vision is, it has only cones. And that's why we like to focus the light onto our fovea so that we can see color well. Now, what's interesting about cones is there's really only three basic colors of cones. So rods, of course, are going to see black and white and gray, but the cones are going to see what we call primary colors. And the reason we call them primary colors is because they're the colors that these cones typically respond to. Now, you can notice that there's kind of a range. When we say blue, we've got a range from purple even into green a little bit. And when we say green, we've got, a bit of a range there as well. So even though we're giving each of them one color, they can be triggered by a range of color, but you can see that their peak stimulation is in, for example, the blue wavelength, the green wavelength, and the red wavelength, or even orange almost. And so that's where their names come from. So the combination of which of those cones, how many of those cones were stimulated, this overlap is what actually helps us see, 1,000,000 colors or 1,000 colors, is because we can get that information from different cones, and how much of each one was stimulated will help us see where on the color line that wavelength is coming from. So cones give us more acute vision, sharper vision, and color vision. Okay, I wanted to talk just quickly about visual fields. It's kind of the last thing we'll talk about here. Visual fields are basically showing, if you're just looking straight ahead, what can you see? What will actually enter your eye and hit your retina and eventually make it to your brain. And so you can have a right-sided visual field and a left-sided, obviously. And even within the right and the left, you can have what we call nasal, which would be the inside on the right, and temporal, which would be the outside on the right. or nasal on the left and temporal on the left. And what you can see is that each eye is taking some information. It's largely getting information from its side, right? So the left eye here is going to get most of its information from the left, but notice it does get some photons bouncing from the right, and the right eye is getting a lot of information from what's on the right, but notice it's also getting some information from what's happening on the left side. So each eye is getting some information from both sides. And then notice what happens as the optic nerves travel from the eye to the brain. There's a crossing at the chiasm, and notice what happens here. You get these kind of temporal receptors here, which were actually getting vision from the right. Okay, they're going to come here and they're going to go off to the left side of the brain. So the left side of the brain is actually going to get information from the right side of the visual field, and that's important. Notice what else is going to the left. If you're looking here at this red line, you're seeing here that it's actually getting information from here on the right side, right? So the right eye and the left eye are going to both give visual information to the left side of the brain. So the left side is going to get information from both eyes, and that's how you can see depth. And the same thing happens on the right. Okay, but notice what happens here at the chiasm. If we follow this red one, which is seeing the temporal, right, the outside of the right visual field, and this green one here is seeing the outside of the left visual field. So you can see at the middle, what is crossing right there at the middle are these outside, or what we'd call temporal visual fields. So the outside vision, the peripheral vision, they cross at the optic chiasm. Okay, once they cross there, then the right brain gets information from both eyes about the left visual field, right? So it's opposite. But the brain can put the information from the left eye and the right eye together, and that helps us see depth. That's where depth perception comes from. That's why if you cover up one eye, it becomes very difficult to be able to distinguish how far away things are. What's the depth with only one eye? We need both eyes to send information to each side of the brain in order for the brain to interpret that information and show us depth. So if we get back to Fatima, maybe you caught it in there in my explanation, but remember Fatima was having problems with her peripheral vision, right? So that's a problem right at that chiasm where the temporal visual information from the temporal side of each eye, it crosses there at the chiasm. So if there's a problem at the chiasm, that will cause a problem with the peripheral or temporal vision. So let's go back to the case of Fatima, and we've got her MRI. And this is a sagittal view here, right? And this would be a coronal view. So if we look on the sagittal view, maybe you can make out some of your anatomy. So here's the pons, here's the brainstem, here's the corpus callosum. So what is this thing right here? That's the pituitary. Now I don't know if you remember, you probably can't remember the size of the pituitary, but it should not be that big. What we're seeing on this MRI is that pituitary gland is quite huge. Now let's go over here to the coronal view, and here's some ventricles, and this is kind of cerebral cortex here. And here again, once again, we're seeing the pituitary, way bigger than it should be. And it's sitting here on the sella turcica. And what's happening is, if you see this, that's the optic nerve. That's the optic chiasm. That's where the two optic nerves are crossing. And so this pituitary is actually squishing the optic nerves. And that's causing those nerves to not function properly. And so she's losing vision at the optic chiasm. And the optic chiasm is where the peripheral vision crosses. So that's why she's having trouble with her peripheral vision, but her central vision is okay. Because the The nasal side of your visual fields, the inside vision, does not cross at the optic chiasm. It goes out in a different direction. So the pituitary is not affecting those axons. Okay, the last thing I want to say is just kind of talk about the coordination of eye movements. So remember we talked about how after the chiasm, you're going to get information that goes first to the midbrain, and those midbrain areas that are receiving visual information are going to help coordinate eye movement. So they're part of the visual reflex to make your eyes move together. And then, of course, the rest of it goes back to the occipital lobe, as we mentioned. And remember, the occipital lobe is getting information from each eye, and that's what's helping you have depth perception. So we will go over some of this in the lab, and we'll test each other's vision in different ways. And hopefully some of this will make a little bit more sense as well. Of course, if you have any questions, we'll have time in lab to go over any questions that you might have as well.